It's great to be here again today with you all, and thank you again for having me. It's also wonderful to get to start to see so many familiar faces that I've gotten to meet over prior conferences and in other countries. So good to see you again. So what we're going to talk about is very much forward-looking, and so these are just some disclosures. The educational purposes is the primary purpose of this presentation, and so we're not offering any kind of precise assurance that this trial is going to start, but we are going to share essentially what we are working towards. That all is going to have to get approved by regulators, by ethics boards, et cetera. So I just want to make sure that I provide that caveat as we get going. You heard a little bit about CAMP4 yesterday, and so I'm going to build on that. We're a group of just over 50 people in Boston, and we are based on a bunch of work that came out of Massachusetts Institute of Technology a little bit over about now 10 years. And the whole orientation of this company is now focused on SYNGAP. So SYNGAP is the lead program, and that's what this group of scientists, clinicians, and drug developers are working on. And our goal is to increase SYNGAP levels with the goal of restoring SYNGAP function and improve disease symptoms. So what we are working towards is a SYNGAP medicine. We're not focusing on any single endpoint, but rather our goal is to restore SYNGAP levels to as near normal as possible with all what we will hope to be the potential benefits that would come from that. And so that's just shown here graphically. You've been through a number of other scientific talks, so I'm not going to dwell on some of the physiology. But just to give a sense of how our technology works, so our job is to increase essentially the protein that comes from the SYNGAP gene. Many of you are familiar with the fact that this is a haploinsufficient disease, and so just to put this in a nice pictorial form, you can see on the left you have a healthy individual, two healthy unmutated copies of SYNGAP. In a SYNGAP affected individual, you see one healthy copy and one mutated copy. And in most individuals, that means you have 50% less SYNGAP protein. And so our approach is to use a RNA-based therapy that targets the control elements, what we call regRNA, and we essentially remove the brakes on the control machinery that tells each allele how much SYNGAP protein to make. And so the goal is to go from 50% to back up to near normal as possible. And this is by telling, in particular, the healthy gene to make more SYNGAP. So we are utilizing that healthy gene. That's the approach here. So what is an ASO? You've heard small molecules, they're gene therapies. An ASO is now actually a type of medicine that's been around for a number of years and has been approved, and I'm going to talk about that in a number of other diseases. It stands for antisense oligonucleotide, and it is a small genetic sequence that is engineered to target a very precise area, and this can be done across a number of different types of machinery. We are focused on the control elements, a regRNA element that tells the SYNGAP gene to make more protein, and so that's what our investigational ASO is doing. And was maybe for anyone who was not here yesterday when my colleague spoke, can you guys raise your hands? I just want to get a sense how much detail to go through. All right, a fair number. I'm going to just run through these real quick just so you can see some of the preclinical data before we get into the clinical trial side. So what we did is we took a mouse model that was actually funded by Cure SYNGAP. At the time it was funded, it was called SRF, but it's now Cure SYNGAP. And so what we did is we took a fully humanized mouse. So these mice have no mouse SYNGAP. They only have human SYNGAP. And we have two versions of this mouse. One has both healthy copies of SYNGAP. The other mouse has only one copy of SYNGAP. So that would be like an affected SYNGAP individual. And so what you can see here is after a single dose, in the orange, on the y-axis is SYNGAP protein, so that orange is healthy, humanized mouse. Then you see in the affected mouse, the gray there is basically showing without any treatment, and then at increasing dose levels, that is getting up close to normal with a single dose of our ASO. What we did then is we put these SYNGAP mice as well as humanized SYNGAP but unaffected mice through a number of tests, and we looked at things like motor deficits, we looked at spatial learning, we looked at increased activity. These are all thought to map to related symptoms in SYNGAP, and what you can see is on the left in the gray, that is the normal humanized mouse. Then the yellow is the affected mouse without treatment, and then the orange is the treated SYNGAP mouse. And what we saw here is essentially return to baseline, or what we call as rescue, across these different domains. We then looked to see, did we achieve more SYNGAP protein in the mouse brain? And what you see to the far right is we got back to 100% in the cortex, and we showed improvement in the hippocampus. And again, this is after only a single dose. So then we went into non-human primates or monkeys, and we wanted to determine, can we also increase SYNGAP protein there? And we wanted to look at particularly at the areas of the brain that we know play a pivotal role in SYNGAP symptoms. And so that's what's done here. We looked at 3 different doses. This is different though from the prior study because we used the route of administration, which means how the medicine would be given potentially in a clinical trial, which would be intrathecally, and we'll talk more about that in a moment. And these monkeys received 3 doses at 3 different dose levels, and what you can see is across increasing doses going from the control, which is the gray, to the darker yellow to orange colors, we see increased levels of protein there. So this gave us confidence that we're seeing it in the right areas of the brain, and we're increasing that protein level. So a little bit more about ASOs. The ASOs have the benefit of the fact that this is a type of medicine that has been utilized before and actually is in multiple approved drugs. And so just to provide that background, we've listed here what you can see is several different types of ASOs that have been approved and given to now, in many cases, tens of thousands of patients. So one of the first CNS ASOs is for spinal muscular atrophy, or SMA, and that's been given now to over 14,000 children around the world. And they receive this medicine at a regular basis. So there's now an enormous amount of experience with how to give it and what to watch for from a safety perspective, as well as the fact that we know it can work across these diseases. So how is this given? This is given via what's called either, you'll hear intrathecal or lumbar puncture. It's essentially the same approach. So the individual lays down, we access the spinal space through the lumbar puncture in the lumbar region, and then we're able to inject that. This for Syngapians is done under sedation, just so everyone knows. And we'll do everything else that we can while we're at it. One of the important things though, actually, I know sometimes this might feel like a bug that you have to, using computer terms, give it intrathecally, but I actually think about it as a feature because what it means is we're not giving the ASO to all the other parts of the body that don't have SYNGAP. Some of it does get into the bloodstream, so what you do have is ASO can go from the CSF in the brain to the blood, but it is much harder, as you may have heard in some other talks, to get it from the periphery to the brain, and you would have to give much higher doses there. So this is actually a benefit is we can get the drug to the right place. A question I often get, and a very important one, is what is the safety profile of other ASOs that are given in the brain? And so there are 2 approved intrathecal ASOs for the brain. There are also many of them right now in clinical trials for Angelman and for other conditions. The common safety events when we looked across is headache, vomiting, fatigue, pain. These mostly come as part of what's actually post-lumbar puncture syndrome. This is a known thing. It tends to occur in around 10 to 20% of individuals, but we have ways of managing it. We have ways of monitoring for it. Occasionally you can get fever or infection. That's one of the more severe risks that people monitor for, and so that's also why going to centers and working with people who have a lot of experience doing these types of procedures can reduce this. In some patients, we will see a very mild elevation of protein in their CSF. This has not been found to be pathologic. It's more an observation and probably comes from some mild inflammation related to the procedure. There are rare circumstances of so-called radiculopathy, which is where you can actually have very severe reaction at the site. And in some cases, children have not been able to walk afterwards for a couple months. And so those are rare, but they are severe. And so it's just important to be aware of these types of events. As I mentioned, one of the things when we look at adverse events is also determining which adverse events do we think are related to the study drug versus what might be related to the procedure or other aspects of the trial. So before I talk kind of forward-looking at what we're proposing to do, I did think it would be helpful to provide some context on overall what do clinical trials and what we often call clinical development look like across other prior now approved drugs, just to give it context, because this is a journey. So it's not a single trial, there's multiple trials, each trial has different purposes, each trial is going to have different populations. And so this is a busy slide, but I thought it would be useful. I'm not going to walk through the details, but highlight a couple points. So this was the development at the point of going into humans for Spinraza, or also called nusinersen. And so what you see was the first clinical trial was a phase 1 clinical trial, started at the end of 2011. It took them a little over 5 years to get their first FDA approval, and that was actually fast. This was through an expedited program. Each of these studies is either a different population. You can see there's a legend down at the bottom. So infants and children, just children across different ages. They also looked at people who had different levels of symptoms. So I provide this just as context to show that any program is going to actually have a number of different studies with different purposes here. This, on the other hand, is the clinical development for Stokes ASO for Dravet. And what you see here is also a timeline where their phase 1/2 took a little bit north of 5 years. And then you can see the phase 2, and they have some long-term extensions that are going on and are just kicking off their Emperor study. So I think it's important, as some of you may think about participating in studies, to be aware of the fact that there will likely be multiple studies in Syngap, and that they will have different purposes that may be a better or less of a fit for you. And so that's what we're going to talk about. We are working towards a potential Syngap phase 1/2 starting in 2026. That is our goal. Before I get into sharing a little bit about the details, the first thing I think is just to touch on a couple, I would say, fundamentals of clinical trial participation. So it's a very important decision, and it's a whole family decision. And so anyone who's thinking about participating in a clinical trial, you want to make sure that you are really weighing the fact that there is both a risk and there is an uncertain benefit. We can show you benefit that has been observed in animals, but particularly the first clinical trials, we do not have any data yet to show that this works in humans. It's also very important to recognize that the different trials at different stages have different levels of burden. That means you may have more activities as part of the trial, you may have more frequent visits, and they may have different levels of risk. There is always the right to withdraw from any trial at any time, and that's very important. So when consent is given to participate in a trial, consent can be withdrawn. At the same time, I think it's important to reiterate, though, that when someone decides to participate in a trial, they are taking a spot that someone else can have, and if they no longer decide to participate in the trial, that can affect the whole community. So it's just important, I think, to think about participating in clinical trial is not something to say, well, let's try it out, and if we don't like it, we're just going to stop doing it. It's a really big decision, and it's a big decision for not just the family, but for the community. I think a couple other things, just in terms of disease-modifying trials, is that these types of trials are very different from, say, more broad anti-seizure treatments and trials. They just have different purposes. And so one of the necessities of these types of genetic treatment trials is we want to get as much what we call homogeneity or sameness in the population, because the population is often going to be smaller, and so we need to make sure that the individuals in any one trial are similar enough that we can observe differences that then we can share with regulators. And so there may be aspects of the criteria for qualifying for some of our initial trials that may not fit everyone. And that doesn't mean that we're not working towards having our treatments be available to everyone or where we think it could have an impact. But it may mean that even if your child or a family member does not necessarily qualify for a first trial, that there may be future trials. And some of the types of trials might be, we're going to look at trials in children versus adults. We learned a lot about the natural history. We know that this is different, whether someone has seizures or whether they don't have seizures. And also different types of genotypes. So do they have a protein truncating mutation? Do they have a missense mutation? These all could impact which trial and at what point someone participates. So the goal of our proposed phase 1/2 is going to be three major things. The first is to evaluate safety and the tolerability. That's always the primary goal of any so-called first-in-human phase 1/2. The second, and this is really important for how we're setting up this trial, we need to figure out what the right dose is in humans. We do a bunch of studies in animals to try and determine what is that right dose, but we don't know for sure. We need to confirm that right dose. And so we need to make sure we have enough measures in as dense a period as possible to be able to select what is that dose for future trials. And so that's going to be a key aspect driving this trial. And then the third is we need to determine, is there some signal that this works? So that is going to be the key three goals. There are several other secondary goals. For sake of time, I'm not going to read through, but that includes supporting regulatory interactions that can enable decision-making, and also support some of the regulatory programs you heard, like Breakthrough. So this is a schematic that is, as of today, what we think is most similar to what we believe our study is going to look like. And I'll walk you through that. But this does have to be approved by every regulator in every region. So regulators may sometimes ask for changes, and so that may occur as we go through this. This is what's called a multiple ascending dose trial. So the way it would work is that a potential participant would be screened to determine if they meet the criteria. If it appears that they do, they go into a baseline period for approximately 4 weeks to confirm and take a bunch of measures. And then people would be randomized to one of 3 different dose levels, but you start in one dose level, and then after that dose level is full, and you confirm safety at that dose level, only then do you move on to the second dose level. So the first dose level would be the lowest dose that we think would still potentially have a benefit based on our animal data. And then after a period of multiple doses, there would be a review of all the safety data. And after that review, if the safety review committee, which is a group of physicians who are part of the trial, determine that they believe that it's safe to escalate to the next dose, then we would initiate the second cohort, and we would do the same for the third. And then after multiple doses, there would then be a follow-up period. And that would actually go out for a number of months, because one of the benefits of ASOs is that they have a very long half-life, which means that they stay in the brain for a while. And that means we don't have to dose as frequently. That also means we want to make sure we measure out to when we start to see that effect start to wane. At the end of the period, the follow-up period here, while it's not shown on this schematic, we would then plan to have what's called an open label extension. And what that means is every individual across these dose levels would be able to elect, if they wish, to go into a long-term study where everyone would get the study drug, and they would get the drug at that dose that we decided was the best dose to take forward. And so that's really important because we're going to look at 3 different doses. And so if we decide that the second dose level or the the highest dose level is the best one, everyone, regardless of which dose level they initially started in, would get access to that selected dose level. So what are some other details? So we think that this study is going to be somewhere between 24 to 60 individuals. I know that's a bit of a broad range, but that's we're working on tightening that up, and that depends a little bit on how many dose levels we end up working. We are also going to have a 3-to-1 ratio where we would have individuals get the study drug, and then we would have, right now, are envisioning having a control. That is part of our discussions with regulators. I'm sure I'm going to get questions afterwards, so maybe I'm going to keep going and then we can talk about a control. But one of the reasons we think it's important to have a control is that many of the scales in SYNGAP are actually taken from parents. And both the clinicians who are part of the trial and parents and family members, we all want this to be a success. But we also need there to be confidence in the data that comes out of this study for regulators. And so that is one of the benefits of having a control, is it gives people increased confidence in the results of the study. The other thing is having a particular genotype. So this initial trial is going to focus on people who have specific mutations. One of the things I mentioned earlier is the importance of picking this optimal biological dose, and that's the dose level that we would carry forward. And one of the ways that we think we can do that faster is by including people who have seizures. That doesn't mean that we don't think people who don't have seizures could not benefit from this. It's rather that we think that that population would be the same or homogeneous. And so that is one of the things that we are planning for in this trial. And there will probably be a minimum number of seizures. But then the plan would be there would be additional studies for people who don't have seizures with SYNGAP. And we anticipate each of those cohorts that I showed you would probably be somewhere between 8 to 12 participants. So as we all know here, SYNGAP affects pretty much every aspect of being a human. And so we need to look at pretty much every aspect of being a human in this trial. So even though I mentioned seizures might be an important part of this trial, we're going to look at a number of things. So this is just a list of some of those types of assessments or domains. You see, yes, seizures, sleep. We want to look at motor and gait. We're going to look at communication. We're going to look at behavior. And then we're going to look at multiple aspects of development. We're also going to look at things like gastrointestinal or GI health. And then there'll be some general assessments. One of the things we're also doing is we are working very closely with the different groups that are part of the natural history studies that you've heard about. And we are looking to use the same assessments as much as possible. So that allows us to actually compare to that natural history data. So that natural history data is very, very important, and much of this study design is based on looking at that data and what we know. So thank you for your participation in those studies. That's very important, and it's really helpful in giving us the highest probability of success. This is also a schematic that essentially attempts to give a little bit of a sense of what the structure might be, but more from a patient visit perspective. So phase 1-2 trials tend to be very intensive. As you get to later stage trials, it's easier to do more things at home. Given the fact that this is done as an ASO, we are going to have people come into the site. There is going to be a real effort to do everything we can at home whenever possible, because we understand that the participation is a significant amount of time and effort. And so you can see the baseline period. There would be multiple doses. Each of those doses would involve someone coming to the study site, having multiple assessments done, then likely having assessment of seizures, and then being dosed. And then you can see the follow-up assessments. This is a list of some of the types of scales. Many of these are the types of scales that you already heard about from the natural history study, and so this just highlights that we will be using those. So first off, I just want to thank all of you for the time and all of you for the encouragement and motivation. This is what my team and I are focused on and what we are thinking about and working towards and looking to have start as soon as possible. So I hope you know, definitely feel that every single day is a day that we need to work to, as quickly as possible, get this trial up and running. So thank you very much for your time and for actually trusting us to work to bring this SYNGAP trial to the community.